I often find it helpful to actually compare a closet full of shoes to the nucleus of our cells, and you'll see why in just a second. So imagine this is your closet, and maybe yours isn't quite so organized, but we're going to take this perfect scenario where you have your shoes very, very well organized. So notice that you have multiple pairs of shoes. You have for each type of shoe, you have a right shoe and a left shoe. Now, those are clearly the same type. They belong together, but they're not identical to one another. So those pairs of shoes that you find in your closet are analogous to homologous chromosomes that are found in the nucleus of your cell. So homologous chromosomes have the same genes on them. They're the same size and shape, but they're not identical to one another. You may have different alleles of those genes on each chromosome. So imagine your shoes, pick out one type of shoe. Your right and your left foot, really, they look the same. They might have stripes in the same place, but you can't put your left shoe on your right foot because they aren't identical. So you have two versions of each shoe in your closet, right? The right foot and the left foot. So we have the homologous pairs for each chromosome in our cells. And for humans, we have 23 different kinds of chromosomes, and they're in pairs. We have a total of 46 chromosomes in our cells. So it'd be similar to having 23 pairs of shoes in your closet. Now imagine your little sister or little brother goes into your closet and decides to pull a prank and just randomly takes out one shoe of each type and hides it somewhere else. So now you're left with each type of shoe still, but only one version, a right foot or a left foot of each of those shoes. So that is similar to the cells or the nucleus of our gametes, the egg or the sperm. They have only one copy or one version of each chromosome. So when we have two copies or two full sets of chromosomes, when we have the right foot and the left foot for each shoe, we call those cells diploid. The DI refers to two. So diploid cells for humans have 46 chromosomes in them, or two pairs, two pairs of 23 homologous chromosomes. Now, when we had that situation where you had only one shoe of each kind in your closet, that is analogous to your gametes, your egg or sperm, which are haploid. So in humans, the egg and the sperm only have 23 total chromosomes because they have only one of each type. Let's look at this in a little bit more detail. So, like I said, your homologous chromosomes are not identical. And you have in your cells 23 pairs of chromosomes. So you have 23 homologous pairs of chromosomes in every single one of your cells. 23 of those chromosomes, or one set, came from your mom, and one set came from your dad. So they're not identical. They do carry all the same instructions for the same things, for the same traits, but the alleles might be different. So you get one allele or one version of every gene from your mom and another allele from your dad. So they both have instructions for making a nose, but those instructions are likely slightly different. Now, 22 out of the 23 pairs of chromosomes we just call autosomes. They are coding for stuff that everybody has. The 23rd pair, and you learned this way early on in the year, the 23rd pair consists of the the sex chromosome. So they do code for some things that everybody needs, like how to make hemoglobin in your red blood cells, or how to make the cells that register color on the back of your eyes. But they do also code for what makes us biologically male or biologically female. So I know this is cut off here, but males, and again, you've learned this, but males receive an X chromosome and a Y chromosome. Females receive two X chromosomes. So DNA organization, before a cell can divide, part of that cell cycle during interphase is the S phase, and that's super, super important. What's happening during the S phase is DNA synthesis. Each chromosome is making an exact copy of itself. So this results in what we call a duplicated chromosome. Now, most of the time, our chromosomes are not duplicated. So the normal state for our chromosomes is this unduplicated state. So imagine you have like a history textbook. There's lots of information in that book. Now, if you carried around two identical copies of that history textbook, it's bulky and you're not gaining any new information from that. So most of the time ourselves, there's no reason for our chromosomes to be duplicated. The two identical molecules in a duplicated chromosome are called sister chromatids. So over here on the right, you can see the two sister chromatids. They are held together by a centromere. And my centromere, I think, is this yellow thing. It moved, but it should be right here connecting. It's a bunch of proteins, protein complex that holds those two identical copies of that chromosome together. Again, sister chromatids are identical to one another. They make up a single duplicated chromosome when they're held together with that centromere. So over there on the right, you can see I said one chromosome because it's held together by that centromere. Attached to each other, they're attached to each other at the centromere. These are not the same as homologous chromosomes. Homologous chromosomes are not identical and they're not held together by a centromere. So whenever they are attached by a centromere, we call those two sister chromatids a single duplicated chromosome, or they make up a duplicated chromosome. When sister chromatids separate, and they're going to separate both during meiosis and during mitosis, we call each of those sisters then one chromosome. And I wonder if I can get there we go. So we call each of those one chromosome. So again, they are identical to one another, but now they aren't held together. So now they get to be their own single chromosome. So if we look here, we have two pairs of homologous chromosomes. So over on the left, notice how those two chromosomes are the same size, same shape. They have the same genes in the same location, but they have different alleles. Same thing with that pair of homologous chromosomes on the right. Same size, same shape, same genes, just different alleles. Each of those chromosomes that we're looking at is made up of two sister chromatids. So these are duplicated chromosomes. They are ready to undergo cellular division. All right, we are almost there. So you can see that diagram on the right. Now, in your head, I guess, think about where is the centromere on these chromosomes. So think about where those should be. I'll go ahead and draw those in and see if that's where you would have put them. So the centromere is holding together the sister chromatids. So we have 4 centromeres, and I just colored them in purple. It's a little hard to see. Okay, we're also supposed to label two sister chromatids. Where would you label that? So the sister chromatids are making up each of those duplicated chromosomes. So they're being held together by the centromere. So here's a sister and here's a sister. So I could do that on each one of these chromosomes. All right, so each chromosome is made up of two sister chromatids. All right, now I'm asked to circle a set of homologous chromosomes. So the homologous chromosomes will be similar size and shape. So if I start with this one, the other chromosome that's a similar size and shape is this one. So these are homologous. So one is the maternal chromosome coming from the mom, one is the paternal chromosome coming from the dad. So they're not identical to one another. All right, in this picture here, I want you to think about, is this showing mitosis or is it showing meiosis? This is actually a fairly tricky question. So we have to figure out what's happening here. What we can see is all four of those chromosomes are lined up in the middle of that cell. And I do have homologous pairs within this cell. So I actually have two sets of homologous pairs. So this cell, I know because I see the homologous pairs, this cell is diploid. So what I'm seeing here is it's a diploid cell, and what's happening, we're at metaphase. What's going to happen next is anaphase, and those sister chromatids are going to be pulled apart to opposite sides of the cell. So sister chromatids are being pulled apart. That does happen in both meiosis and mitosis, but the difference is in meiosis, the sister chromatids don't get pulled apart until meiosis 2. At that point, the cell is already haploid. So I know that this has to be mitosis because I have a diploid cell that's going to be dividing into two, and the sister chromatids there are being pulled apart. We can't have a second division after that.